So, hello everybody. I am Luca Rima. I'm a PhD student from the CESINA at the University of Basel in Switzerland. I'm a member of Thomas Brown's group who should have given the talk today, but unfortunately cannot be here with us today. So I got the opportunity to talk to you about our microfluidic sample preparation device we call the cryoWriter. So in the next few minutes I will try to give you a rough idea what is the cryoWriter and what it is capable of. So our cryoWriter system is built in a very modular way, and this is an overview of all the modules we already developed or which are under development. And I will give you now a quick explanation to all of them, and later on I will go more into the details. So on one hand we have a part which is dedicated to single cells, so we can monitor and lyse single cells and prepare the cell lysate for EM analysis. Then we have a part which we are developing at the moment, which is for cross-linking, where we hope to stabilize fragile proteins and complexes prior to sample grid preparation. Then we have two parts for protein purification prior to grid preparation. One uses an electromagnetic trap combined with superparamagnetic beads, where the protein is bound, retained, and then written onto a grid. And the second purification method is capillary electrophoresis, where the protein can be separated before the grid is written. Then, of course, we have a part for plunge freezing, for sample vitrification, and the conditioning module, which we use mainly for negative stain EM. And a handover system to connect all of those modules together. The important part you should keep in mind is that all these different modules make use of the same microcapillary. And this has several advantages, like minimizing the sample interface contacts, the loss by unspecific absorption, as well as the tailored dispersion. So, as I just mentioned, the system is very modular. So you can do many different experiments by choosing the modules you need for your experiments. And I give you now some examples from experiments we are doing quite often in our lab. So for negative stain, for normal negative stain, you would just use the conditioning module together with the handover module. For protein purification by fishing, the module with the electromagnetic trap, then the handover, and then the plunge freezing for vitrification, and then go to the EM. Then for visual proteomics experiments, we would grow the cells in our setup, monitor them, lyse them, and prepare the lysate for the EM, in this case negative stain, but of course it would also be possible with cryo. And you can also do experiments not related to EM at all, like preparing single cell lysate for reverse phase protein arrays, as well for liquid chromatography and mass spectrometry. This is actually how the setup looks in real life. So it consists actually out of many high precision linear stages. We have a high precision pump in there, which gives us sub-nanoliter precision to manipulate the sample. This syringe is connected to the capillary, which is now somewhere here. Then we have many cameras to monitor what is going on. Here is the inverted fluorescent microscope with the cell incubator on top. Back here in the back you can see the electromagnetic trap. On the right side we have a dew point stage, which allows us to control the temperature of the grid and produce a microenvironment around the grid, which is important during grid preparation. And here on the right hand you see the plunge freezing mechanism for sample vitrification. The whole system is controlled and programmed using OpenBEP. This is an in-house developed development framework. OpenBEP stands for Open Biological Experiment Browser. And this software allows us to develop new modules outside the system, and when they are finished, integrate them later on into the finished system quite easily. And also all the steps done during an experiment can be automated using the macro language of the software. Now to the details of the different modules. First to negative stain and cryo. So there are some major differences between our method and the classical sample preparation methods you all know. So first we are using much smaller sample volumes. So usually for a grid we use between 5 and 20 nanoliters of sample. And as well we are virtually lossless, meaning that we have no paper blotting step included in our procedure to making a grid. So you know, if you have the paper plotting in normal sample preparation, you lose more than 99.9% of your sample, and also it's a rather harsh treatment, this this paper plotting step. So we have no need for this. The negative stain in more detail works as follows. So the first step is sample uptake, where you withdraw several nanoliters of your sample. This is followed by a conditioning step where the sample is stained and desalted at the same time, and then it's deposited on a cooled grid and dried very slowly. And here in the corner you can see an overview picture of a grid, and if you look closely here you see the area where the sample was written onto the grid. The conditioning for the staining I just mentioned relies on the fact that salt ions and stain, here it's a tungsten-based stain, they are diffusing much faster than proteins. So if you just condition for a few minutes, you see that most of the salt is going out of the sample plug, the stain is going into the sample plug, but most protein still remains within this sample plug. And these are some example pictures. So this is an overview picture of the whole droplet on a slot grid. So we sometimes use slot grids to have accessible the whole sample in the EM. So no part of the sample is hidden behind grid bars. And this in principle also enables quantitative experiments and stuff. And those are two example pictures of TMV, and you can see the staining you can achieve with this conditioning technique. Now to the cryo part. This one also involves 3 steps. So the first step is the sample priming, where the sample is written here on the grid in a spiral pattern, and in the end all the excess sample is removed. This grid is controlled, is temperature controlled by this dew point stage where it is always in contact. This is followed by a very short thinning step which can be monitored by laser absorption, and then it's rapidly plunge frozen in liquid ethane for vitrification. So this you can see now here how it looks like. It's a bit slower the way than in real life, but the capillary is approaching the grid. Then the sample is written onto the grid in a spiral pattern, and you will see in the end the capillary is moving back to the center, is withdrawing the excess, and then it's rapidly plunge frozen. And then afterwards you will see the plunge freezing in super slow motion in a side view. Now, yeah, and then it should be nice and fine, hopefully. So these are some example pictures. Here it's an overview picture, and if you look closely here, you can see the borders of the droplets, and within is the area with vitrified sample. The middle picture should give you an idea about the homogeneity of the ice within those squares. And on the right side, you can see a picture of a sample containing epoferritin and phages. Now to the purification parts of the setup. So classical protein purification tends to use rather high volumes and quite a lot of time. But in the end, for structure determination, you only need several thousands to millions of particles. And this amount of proteins can easily be handled by microfluidic techniques. So this classical pipeline indicated here is in principle not needed. So we try to integrate this also into our system, and this we were doing by installing an electromagnetic trap in the system. So this one is consisting actually out of 2 electromagnets with 2 cooled iron tips, a camera for monitoring what is going on, and a UV LED for photocleaving the crosslinker, so for photoelution of the protein in the end. And the capillary is indicated here in between those 2 tips. The function is as follows. So your target protein is bound by a Fab fragment, and this Fab fragment is connected to a superparamagnetic nanoparticle by a photocleavable crosslinker. If you now want to fish a protein, first you are going to do is creating a plug of the beads within the capillary in the trap, and then flush over a mixture of proteins, which is containing your target protein, obviously, which was pre-incubated with Fab fragments and crosslinker. Your target protein will then bind to those beads, will be mobilized here in the trap, and all the rest is flushed away. This bound, your target protein can then be written directly onto a grid, and a superparamagnetic nanoparticle can be used as an electron-dense label, or prior to grid preparation, it can be cleaved off by the UV LED, which is also built into the setup. These are some example pictures, how this could look like. So the structure is actually some structure related to malaria we got from the Tropical Institute. So these are two pictures in negative stain. Here it was not cleaved, the beads, so you can use it for tracking if you like, or you can also cleave it off and then it's gone. And here it's a cryo picture, and here you can also see this structure. This one was also with photo illusion, so with removal of the beads. And now this is a very interesting project from a former PhD student in our lab. So what he was doing, he was buying commercially available HeLa cell lysate, and he was using 900 nanoliters of HeLa cell lysate as starting material. And by using this trap, he was able to fish the endogenous native human 20S proteasome and making two grids. And this is how one of the squares of this grid was looking like, and this is an example picture which was taken in here. And you can see on one hand the proteasome he was fishing, and on the other hand you can also see TMV, which he was adding as a resolution control. And yeah, by just using this 900 nanoliters of starting material in fishing, he was able to get the structure of the endogenous human 20S proteasome and reach the resolution of 3.5 angstrom. Maybe here it's also worth mentioning that the human 20S proteasome only has C2 symmetry and not D7 as in many other species. The TMV, on the other hand, which was added as a resolution control, we got the final structure at 1.9 angstrom resolution, with some nice densities, even showing the holes in the aromatic rings. Now to the second sample purification method we have built in the system. It's the capillary electrophoresis. So in capillary electrophoresis, the proteins are separated due to different electrophoretic mobilities, meaning this is dependent on the charge of the protein as well as on the size. And once again, we are able to do this purification by using the same capillary as we're using later on for the grid writing, which gives us the possibility to work with very small sample volumes, and as on top we are not losing a lot of proteins due to absorption to the walls of the capillary. So this is actually quite new. So we haven't done cryo yet, but a master student in our lab was already performing some initial tests in negative stain. So he was using 10 nanoliters of E. coli cell lysate from E. coli overexpressing apoferritin. And after the electrophoresis, the apoferritin is actually well separated from the rest of the lysate. And in the top fractions also the concentration is rather good. So what we are now trying at the moment is to prepare cryogrids out of this material. Then to the single cell part, as I mentioned in the beginning, we are able to grow, monitor and lyse single cells in our setup and prepare the lysate for cryo-EM as well as for negative stain EM. And the lysis is done by electroporation, which is working as follows. The cells are grown on ITO-coated glass slides, so electrically conducting glass slides which are grounded. And the electric field is generated by a function generator which is connected to this platinum-coated capillary. Then after this electroporation, the cell is immediately withdrawn into the capillary, and by the present shear force it is lysed completely. This is how it looks like in the fluorescent microscope. So here is the cell before lysis, and here it's after lysis and uptake. And the shadow you see is the capillary seen in the microscope. And those are some example pictures, one in negative stain, one in cryo, and you can nicely see cellular structures like filaments, membrane patches, and also some membrane proteins up there. And what another PhD students of our lab is now doing is we developed this procedure we call differential visual proteomics, where structures from different cells are classified and the abundance of the found structural classes are then compared to find differences in the proteome. So in this schematic here, a negative and a positive control is classified, the structures are compared, and here, for example, you would have found a difference in this ring-like protein structure. Then another technique we are using for getting more information about protein and the interaction of the proteins within cells is this interaction labeling, where we are using a fab fragment with a superparamagnetic nanobead for one protein and an electron-dense label against the potential binding partner. Combining this with the electromagnetic trap and the grid preparation methods we developed before allows us to label interaction partners, wash away unbound labels and unbound proteins, and then write those labeled interaction partners onto grids and analyze them in the EM. And this can then look like this. So what was done here was, in the first fishing step, apoferritin was fished out of cell lysate. Then in the second fishing step, this apoferritin was used as a bait for ferritin antibodies. And in a third step, these ferritin antibodies were labeled with immunogold. Then the magnetic nanoparticle was cleaved off, and then the remaining stuff was written to a grid. And this you can see here. So the arrowhead is actually the immunogold, and the arrow is pointing to the antibody. And the antibody served in this experiment as the binding partner. So, to summarize this all quickly, the cryo-writer we developed is able to produce grids for high-resolution cryo-EM. The sample purification methods implemented allow us to purify samples right before grid preparation with small amounts of sample. The possibility to grow cells in the setup and to prepare the lysate for EM allows or enables single-cell visual proteomics experiments, and the interaction labeling can give you even more insights about proteins and the proteome, the interactome of a cell. And currently, at the moment in the lab, we are working on a cryo-writer 2, so on a new version of the cryo-writer with even more possibilities for grid preparation, and we are planning to bring a simplified version of the setup to the market. And the company, which is the startup company which is working on this, is called Nuonex, and if you want to have more information about that, you can visit the homepage nuonex.com. And yeah, last but not least, I want to thank all the people who were involved in these projects, especially my two bosses, Thomas Braun and Henning Stahlberg, and of course also all our team at CSINA. And all of you for your attention, and if you have any questions, I would be glad to answer them now.
Many thanks, very interesting. So we have really plenty of time to discuss. It's on purpose. We have 20-minute talks and 10 minutes discussion. So take your time and go for it. Albert, maybe?
How long does it take from picking up the grid to after you've written your sample to the grid to freezing?
At the moment, I'm using 360 milliseconds at the moment.
But that's sort of that's fixed. Or could you vary that?
It's fixed. It's very. At the moment I cannot go much faster, but I can go much slower.
Sure.
But I am thinking on adjusting some stuff on the motors, and yeah, then I can go also faster. But at the moment I fixed it at 360 milliseconds. With the current setup, this is what is more or less possible. But it could be better, but then I need to improve some stuff with the motors first. Yeah.
But then it's still interacting with the air sometimes.
Yeah.
You cannot avoid that.
No, not completely, no.
But the total amount of liquid you really need, because you start spreading and then you soak it away, right?
Yeah, so I put 20 nanoliter on the grid, but in the end it's much less, right? So in the end, if you would have 100 nanometers over your whole grid, it was 900 picoliters or something like this, right? So in the end, there remains much less, yeah.
One delicate issue when you do it now in the right robot, the samples, is the thickness of the grid. Now I saw in your scheme that in one moment you adjust the thickness before doing the quick flash cooling. How can you precise adjust this thickness
Mm.
mechanically?
So first thing would be with the laser, you can measure the laser absorption, which gives you an indication of how thick it maybe is. The other thing is just by, you try to withdraw as much as you can, of course, and then what is pretty crucial is the temperature, which the grid is. So you control the temperature of the grid, and depending on the dew point which is around the grid, this gives you a small evaporation, and by tuning this evaporation rate, you can then also tune the thickness of your final sample. Yeah.
But it means the concentration could vary during that process.
Yeah, there is a small amount of evaporation, not a lot, but yeah, of course, the concentration is going a bit up there, for sure. Yeah.
Yeah, just so we get prepared already, do you have a rough number of the budget we would need?
It's the startup is now in discussions with some engineering bureaus, so I cannot give a rough, precise number there. But for sure, it would be much cheaper than what we have in the lab right now, because these motors are completely overkill. So we would go for a much cheaper price than what this setup was costing. But I don't want to give you a number in now, because I'm not involved with the discussions with these engineering bureaus as well.
If you avoid being bought, then it may remain reasonable. That's a good problem.
Yeah.
Hi, really interesting talk. Have you done any comparisons? If you just take, for example, the purified ribosome or electrophoresis or whatever, have you done any comparisons between how much concentration of sample you need for your system versus on a Vitrobox or on a standard fossil environment?
It's dependent on the sample, it varies, but usually we need lower concentration. Especially if your protein tends to stick to the filter paper, you sometimes can really dilute it first with buffer, because otherwise it looks almost like a 2D crystals and sometimes. But it's really highly dependent on the sample, but usually lower concentration for us than what you would use on a Vitrobot. Yeah.
That's really interesting, because I think colleagues who've used systems like the Spotterton have reported that for the Spotterton, for example, you need slightly higher concentration versus nods compared to the Vitrobot. So can you speculate as to why that would be? Because both of your systems have similarities in terms of applying particular volumes onto the grid.
I would just be speculating. The Spotterton is still a bit blotting by having a self-blotting grid, so maybe it's the flow within the squares, or maybe it's just because I have a little bit of evaporation, not a lot, but it maybe also drives a bit the concentration there. But it's just what I'm seeing. I have not looked into it like in a very scientific way. It's just my experience that I see more particles usually if I compare to the Vitrobot.
But if you compare it, I mean, if you take a reference sample and do a classical grid preparation and with yours, how is the particle concentration looking afterwards?
Yeah, the concentration is high in most cases, but this is sometimes it's a lot, sometimes it's almost the same. I think it's really dependent on how much protein you lose in the filter paper.
Okay, because a couple of us here did a test last week with the Spotterton, and actually you need really a lot more. Otherwise you have hardly any particles.
No, I have the other experience the other way around.
Did you try to do some tomography to see where the particles are sitting in the film?
I'm planning to do this, I haven't done, but it's planned, yeah.
And can you can you control the ice thickness somehow, by waiting more time or?
Yeah, by waiting more time for sure, but this also drives the concentration a lot. So what you try to do is really have it as short as possible, to have air-water interface for a really short time and not a lot of evaporation. But I think the thing is really how you spread it, how you do the glow discharging, how you withdraw the sample. And then by choosing the right offset temperature for having this little bit of evaporation to get to the right thickness. Yeah.
There's the first question with the microphone.
I'm honored. Do you glow discharge the grid outside of the system, or do you have a glow discharge?
Yes.
Does it depend on the glow discharge house? So I guess it depends, so you can control how it spreads the liquid with the glow discharge. No?
Yeah, it makes a difference, but normally I'm using normal glow discharge now. I was doing some experiments without Solaris to use different plasmas and I don't know what, but in the end I was going back to the normal glow discharging. But it makes some difference, yeah. And it's maybe also depending on your sample, you have to check what is best. Yeah.
How long does it take to write the whole grid from the beginning to the end? And how big is the usable area?
The usable...
The usable area, if you can use the whole grid or just
the whole grid would, I don't know, it's a few seconds of writing, I don't know, maybe 3-4 seconds, and then plant freezing within, after filling up to 360 milliseconds, you plant freeze it. And then if you have a good grid, normally you have the droplet, and then in the center part it's a bit too thick, and then it's a rim where it's good ice, and then it's a rim where it's too thin, the ice. And then I have maybe, maybe perfect 20 squares usable, which is enough if you have multi-shot on your Titan for one grid for a lot of data collection. Yeah. And if you have a bad grid, then maybe only 3/4 grid squares are with usable ice. Yeah. But this needs to be tuned. So best is like you have first a screening day when you do different conditions, and then you check. And if you have good conditions, it's reproducible. Yeah.
Have you tried it in a cartridge, in a Theos cartridge? So if you have the grid already loaded in a ring.
Yeah, but not me. I was another student, but by plunge freezing it's hard to really have nice vitrified ice. So for having clipped grids, maybe you need to have this ethane jet or something.
So, some more questions?
Can you go towards time resolved freezing, that you really are able to have a time series, or is it too long already?
Longer I can go for sure, but shorter than this 360 milliseconds, as I said, it's hard at the moment. Then I need to adjust, but longer for sure, yeah. Longer I can do immediately, and then just having the grid at a dew point, so there is no evaporation at all. This would be possible, yeah. But shorter, not at the moment. Yeah.
I have a question related to the purification via the microfluidics.
Mm.
So, these, I mean, it looks terrific, first of all, but these appear to be fairly abundant molecules, like the purifying of complexes.
Mm.
Have you done any tests to see, say, you take a lysate from E. coli, human cells, whatever have you, spice something in? When can you actually pull it out versus background?
So actually the proteasome I showed you was endogenous, so this was commercially viable cell lysate. This was not overexpressed, nothing. Now we are trying with more rare proteins than the 20S proteasome, but like we are still in testing, so it's hard. So I cannot tell you exactly the concentration which you need, but you can in principle also just use like the overexpressing E. coli already and lyse them, and then just use a very, very small amount of this lysate, and then for sure it's enough. Yeah.
And so you're thinning, what is happening exactly during the thinning step? Because you showed a beam coming, but this is not thinning itself.
No, this is just for monitoring. So what you do first, you do a blank with the empty grid, and then you know what is completely dry, and then you can measure the intensity is much lower if you apply the droplet. Then after withdrawing this little bit of evaporation, you can see the intensity is coming back, and then you can set the threshold when it should be plant frozen.
It tells you immediately whether the grid is good or not, which is also quite interesting.
More or less. The problem is that the quantifoil grids are not always super... They are not always the same, so they have sometimes a different coating, so you need to do this blank all the time, and then sometimes this value changes also a bit. But it's, if you have a good batch of equal grids, it's working very good.
But the thinning itself is just the waiting step, or
it's the withdrawing and then the small waiting step. Yeah.
Okay. There's one more question there.
You were talking about an ice distribution around the grid. Do you see also around inside one square or inside different squares if you have an ice distribution in a square, or is it
like different ice on the grid?
Exactly, different.
Yeah, you have a gradient. As I said, normally in the middle is too thick, and on the outer rim it's much too thin, and in between you have a gradient.
Yeah, this is on a grid level, but what I'm talking is more
within one square.
Yes. So when you do the GP2 or Petrobot, then you mostly see a difference on square level.
Or like close to the bars, there is sometimes it is thicker, but
okay, but it's not
super close to the
so
bar,
that you
it's
see
usually
it everywhere. Okay, good. Thanks.
Because you're relying on evaporation primarily for your thinning step.
A little bit, not a lot, but
yeah.
yeah, well, we need a bit. Yeah.
But even a little bit of evaporation could lead to changes in or concentration of buffer constituents, which might not be a problem for lots of proteins, but for the more labile complexes, which are increasingly the ones we're looking at, they might cause some problems. So have you seen any evidence of, for example, salt concentration?
I see if it's going wrong. This I see. If I was waiting too long, I can clearly see that the salt concentration gives me a high background noise. But if everything works fine, it's okay. But if I wait too long, I see it in the microscope that there is a background. What can be done? What I was doing in the past sometimes is the whole system is preloaded with water, because if it would be air, like you cannot really manipulate nanoliters, right? And then you can just also, after putting the sample to the grid, just add additionally a little bit of water to compensate this evaporation. Just maybe 1-2 nanoliters after your 20 nanoliters of sample. And this can maybe already help you in some cases to compensate this increase in concentration.
Sorry, I'm quite interested about electrophoresis part. Can you just explain a little bit in more detail how it works, the principle? If you talk about it.
Which part?
Electrophoresis.
Ah, okay. Yeah, in principle you have the capillary and then you have 2 reservoirs and then you put a high voltage there and then you keep cool the whole capillary and then the protein starts moving. And the capillary is passivated, so you don't have a flow in there, so they should just really move depending on their size and their charge. And
so different proteins have different charges.
Yeah, and different sizes, so they move at a different speed. And then they will separate over this capillary. And then you need to do first a test, maybe negative stain, to see after how many seconds your protein is coming. And then, if you know the time your protein is taking, your target protein, you can then do the cryo-ERIC if you know now it's coming then. Or you can add a detector, but the one we had broke, so at the moment I'm stopping the time. So.
Have you thought of combining that maybe with fluorescent markers to identify, if you take a lysate, you could also just start it directly in your complex? Is that something?
We were thinking about this, but it's quite new, this electrophoresis thing. Yeah. It was developed the last half year, so.
That's interesting to use with small volumes also.
You just, sorry.
That would be.
You're just talking about the detector you broke. What kind of detector you used?
I was.
Can you repeat the question?
What kind of detector you were using?
Yeah, he just mentioned about detector they have used to detect, but it's broke. I
don't know. It was a laser, and then the detector was measuring some fluorescence, I guess, or some absorption. I don't know. Actually, the laser broke. I was never using this one, but I think it was. UV, maybe just a UV laser, and then they were measuring the absorption or something similar to maybe the nanodrop is doing with this UV stuff. No? I have to check. I cannot tell you exactly which one.
Yeah, back to the writing on the grids. So if I look at quantifolds in an SEM, you basically see that the grid square is a little bit of a well.
Yeah.
Because they put the carbon on the round side of the grid bars, whereas Proteochips puts it on the other side, on the flat side. Have you tried both types of grids, and do you see a difference in how...
Between quantifoil and lacy, or what?
No, quantifoil and Proteochips, C-flat.
C-flats. I use both. Most of the time I'm getting the sample from somebody and they tell me I want to have those grids and I'm just using those. But yeah, I was using both and I have different experiences with them, but both work fine. Both work fine in principle. What I have seen lately is that sometimes the proteins tend to stick more to the carbon on the C-flats, and I don't know why. If it's because, but I don't know. I haven't done, most of the time I'm using anyway quantifoil. So 95% of my experiences with quantifoil grids and C-flats I'm using if the person is like, yeah, there is plastic on the quantifoil, don't use this, and then I'm using the C-flats.
Maybe they're just more hydrophobic in the beginning, no?
Possibly, yeah. I have to make some experiments maybe on this, but I was not looking really systematically into this problem. I'm more working on my machine and just taking the grids which are around.
And the thinning, if you're waiting that your sample is sort of slightly drying, let's say it was spreading and slightly drying, so the drying itself will not be linear, no? Because the thinner it is, the faster it will evaporate.
Yeah.
So is that something you take into account somehow, or is it just you measure and try?
I measure and try. I do different conditions and then I do screening. And in the meantime, I have some experience. And even like there are cameras, just with the camera, you already see sometimes, okay, this one was completely dry or this one was much too thick when it was done. So after doing hundreds of grids, you can even see in the camera and by feeling and already tell like, yeah, this one was a good one.
Because what you mentioned that you add a bit of water at the end, if it's a complex, a multi-subunit complex, and your salt concentration is rising, then it may be too late to add water. It's already saving. So that can be quite delicate. Is there a way to avoid that completely?
You can in principle do the grid exactly on the dew point, have no evaporation at all, but then it's hard to really get the nice thickness of the ice. So this is just by withdrawing, it's a little bit too thick normally, so you need this small amount of evaporation. And I was also thinking of maybe adding some other stuff to solve this problem. This is
so a two-part question is, let's assume you have a sample and you want to make several grids. How long does it take per grid? And then the second question, assuming you have several users with different samples, how long does it take to wash the device in between and start all over again?
So if the whole setup is set up, so it takes me just like 2-3 minutes per grid. So to set up all positions and prepare the ethane, so this takes half an hour for sure. But if everything is set up and you saved your positions, then it's like 2-3 minutes per grid. And washing the nozzle, so usually I just flush it with ethanol, or if you want to go very sure you can use.
So it means after an hour or so the next user could come and use
it? In principle, yes. At the moment I need to use the setup myself because it's a prototype, so I cannot just give it to users because it's not crash safe. So if you press the wrong button, it's broken. Huh?
You're trying to commercialize it, so I
yeah.
would like to
So
understand.
then it would be much more user friendly than it is right now. But in principle, setting up half an hour freezing grids then takes like 2-3 minutes per grid. So the washing and setting up a new sample takes then, if the ethane is already there, maybe 10 minutes, 15 minutes. So.
How is that condensated actually?
How is it?
Condensated, or you control the temperature for that also?
By Peltier. So normally by Peltier elements.
And do you measure it also? Or?
Yeah, I measure it. Okay. Yeah. I have several sensors in there for measuring grid temperature, air temperature, humidity, temperature of the capillary. There are many sensors in there.
There was a question in the back as well. I forgot about it.
I like the modularity of the system. You can add many modules. Do you think gel filtration will be an option, and then you will be able to have a two-way junction where you select just one fraction along the profile?
I never thought on this, but if we have time and man, maybe it's a good idea. I have no time at the moment for this as well, but maybe it's a good idea. But I was never thinking on that. And we started with the electrophoresis, so we try to do this first, but it's a good idea. Maybe. I have to look into it.
And maybe how can you have experience on how effective it is to capture low abundance protein in the purification module?
How efficient it is?
To capture low abundance protein in the cells.
How high is the efficiency? So we are working now on some proteins which are much less common in the cell, but we have no results there so far. So what worked, this was like half a year back when we published this, was the 20S proteasome, and this is what we are working on at the moment to get also like much more proteins out of cells.
Sorry, just quickly. And non-brain proteins?
Like the writing or what?
No, can you, how your purification would work? Are you trying non-brain proteins?
I think one student of us is working at the moment on the membrane protein, yeah.
Yeah. Is he happy?
Huh? I think he has actually, he was doing his grids last Friday, so I think he's looking at them today or tomorrow. So yeah, fingers crossed.
Yeah, sounds